Today we're going to be talking about protein synthesis, and this is a process which is basically how proteins are created. And there are a lot of organelles involved in this process, which we are also going to be talking about. This is lactase, and this is an example of an enzyme that would be created by protein synthesis. And what happens is this journey begins in the nucleus. The nucleus is a membrane-bound organelle. It contains DNA, and from this DNA, RNA is created. So there are three types of RNA, and these are messenger RNA, transfer RNA, and ribosomal RNA. And our DNA is basically a long series of instructions for how the amino acid should be arranged into proteins. However, the instructions are encoded in such a way that they have to be translated to be read, and the DNA instructions are transcribed into mRNA, which can then be decoded by ribosomes. Ribosomes are made up of two subunits, a larger and a smaller subunit, and these are both made up of rRNA and proteins, which, as you recall, is the ribosomal RNA. And the subunits normally exist separately until the small subunit binds to a certain mRNA message and then sends for the large subunit so that they can form the complete ribosome. When these two subunits come together, there is an mRNA decoding site in between them. So this is where translation occurs. Translation is the process where proteins are created from mRNA strands. Now, this is done by matching the correct tRNA sequence to the correct mRNA sequence. Now, if the ribosome hits a specific sequence of mRNA, it realizes that the protein needs to be synthesized by the rough endoplasmic reticulum, which we will talk about next.
I'll be talking more about the endomembrane system. The endomembrane system is the process of protein synthesis from transcription within the cell to the distribution throughout the cell. The next step in the endomembrane system after transcription takes place in the endoplasmic reticulum. The endoplasmic reticulum, or ER, is a structure of complicated folded sacs and compartments. It makes up a key part of the endomembrane system, which is the system by which proteins are synthesized. There are two parts to the ER, the smooth and the rough. The rough is coated in ribosomes. These play a crucial role in protein synthesis. The rough ER is where the information from the RNA is synthesized by ribosomes into proteins. This process is otherwise known as translation. The proteins enter the RER lumen, or interior, and are chemically modified or folded to fit the specific function of the protein. Once the protein is fully synthesized, it is sent to the Golgi apparatus. Near to the RER, the Golgi is another series of membranous compartments forming a lumen space. The Golgi is responsible for distribution of proteins throughout the cell. It sends them to whatever part of the cell they are required in, or envelops them in a vesicle to be transported out of the cell by exocytosis. Exocytosis is the process by which proteins and waste products are moved out of the cell. The Golgi apparatus forms vesicles, which are small compartments that serve as transport through the cytoplasm. These contain proteins that can't be used within the cell. Vesicles have a membrane of phospholipids just like the plasma membrane. Therefore, they can meld with the plasma membrane and deposit the proteins they carry outside the cell. Exocytosis is also useful because the vesicles add fresh materials to the cell membrane. In conclusion, it is clear that protein synthesis is vital to cell function. Without the fluidity of the endomembrane system, our body processes would not work.
Overall, protein synthesis is a vastly important part of ours and other animals' existences. There's just so much that they do, like from protecting a body against hostile invaders to allowing for movement. Like, this is all done by proteins, and that's why protein synthesis is so important.